Okay, good afternoon, everybody. I'm Deyi Zhang, a senior chemist in the Office of Research and Standard in the Office of Generic Drugs at FDA. I hope you all had a nice lunch break and enjoyed the morning session and also the sessions yesterday. All these sessions were focusing on the immunogenicity risk mitigation strategies and also different methods and assays used to evaluate immunogenicity risk for peptide drugs. Now I want to switch gears to start the second main topic of this workshop, the synthetic oligonucleotide therapeutics. In this session, we have invited experts from both industries and also FDA to share their experience and also knowledge in terms of characterization of oligonucleotide-related impurities, also innate immune response modulating impurities, so-called ERMIs, and innate immune activation by oligonucleotide, as well as immunogenicity risk assessment consideration from both regulatory and also generic industry point of view. Next, I will briefly introduce our speakers. Our first speaker is Dr. Markham, who is the Division Director of the Division of Therapeutic Performance 1 in the Office of Research and Standard in OGD, FDA. Our second speaker is Dr. Yang, who is a Senior Research Scientist at the Division of Pharmaceutical Quality Research 2, Office of Pharmaceutical Quality Research in the Office of Pharmaceutical Quality in FDA. Dr. Yang is going to join us online. Our third speaker is Dr. Bart Rogers, who is a research officer and also reviewer in the Division of Translational and Precision Medicine in the Office of Clinical Pharmacology, OTS, at FDA. Dr. Rogers is also going to join us online. So this session, we have two speakers join us virtually. Our fourth speaker is Dr. Sudhir, Agrawal, who is the founder and the president of RNA Sciences, and also a Filip professor at the Department of Medicine, University of Massachusetts, Chan Medical School. Our fifth speaker is Dr. Hannah Lee, who is a pharmaceutical scientist at the Division of Pharmaceutical Quality Research 4 in the Office of Pharmaceutical Quality Research, OPQ at FDA. Our last but not least speaker is Mr. Filip Kolenc, who is a senior global toxicologist at Sandoz Global Development. So without further ado, I want to invite our first speaker, Dr. Luke, to the podium. He will give an overview of oligonucleotide therapeutics. The title of his talk is Opportunities and the Challenges, Generic Oligonucleotide Drugs.
Thank you, Deyi. Isn't this session going fantastic? I mean, these 2 days, we've learned so much, right? And we've come a long ways from, like when I was at NIH, I was in a laboratory that was trying to break tolerance for a skin disease, certain skin diseases as models. And the tools that we have advanced somewhat. And it's really nice to see that we're trying to apply this to our generic drug enterprise. Of note, some of the new technologies, it's not always clear what role they would play in looking at tests versus reference. We heard, for example, the last session, there were lots of new tools. How would you apply those for tests versus reference? I would challenge the folks that presented those to focus in on that. This is my disclaimer. And of note, for the disclaimer, I had a nice photograph of a pictograph of a oligonucleotide. Of note and relevant was on the 7th, the Nobel Prize in Medicine went to a pair, a duo that worked on microRNAs, which the mechanism behind microRNAs does form the basis for these therapeutics, the oligonucleotide drugs that they do RNA to RNA binding, et cetera. Oligonucleotide drugs regulated by CDER as NDAs and ANDAs are chains of nucleic acid, approximately 20 to 30 to 40 mer for currently approved drugs. There's not been a defined cutoff for size for these products, unlike with peptides. Oligonucleotide pharmaceutics, they work on targeting specific genetic sequences for medical purposes. Usually they're, well, for many of them, they're there to rescue mutations or problem sequences, and you're trying to introduce a sequence fix. They're rapid advances for biopharmaceutical therapeutics and preventatives for previously untreatable disease. There are a wide range of target diseases, and I'm going to get into some of that: genetic disorders, cancer, viral infections, for example. And there's a huge potential for personalized medicine and rare disease, but along with that comes potential for huge cost for the public health system. So some of the economics behind personalized medicine come into play. Here's a list of all of the FDA-approved oligonucleotide drugs since 2023, and you can see it, they fit nicely on one page so far. Eventually, we'll have to move to a second page, but you can see they vary as far as categories. We categorized them by the first ones were phosphorothioate acyls, and then we've got some phosphate oligonucleotides, aptamers. We've got some various other modifications to the chain sequence to enable those products to stick around longer in the physiologic milieu to prevent degradation. You can also see some of the varying lengths of the oligonucleotides here in this chart. I think these slides will all be available on the CRCG website, so don't focus too much on looking at the material, but you can see they range, they're around the 20s, so usually, as far as the length of oligonucleotides. And the approval dates range from 1998 all the way to 2023. And you can see there's a lot more recently than over the past decade. This is the same list, but extended a little bit about the indication. And so you may wonder, okay, what are we targeting? And the indications are generally mutations where there's exon skipping, or there's fixing some aspect of a hereditary disease that you're trying to introduce either suppressing a message or modifying the message as it goes out from the in the cellular protein making machinery, the translation machinery. Many of these oligo products have been approved for rare orphan diseases. Some of these oligonucleotide drug products have been approved on the basis of surrogate endpoints. And of note, many of these have post-marketing commitments to say, okay, well, you need to study this a little bit more and tell us what the safety and effectiveness is. Do look at the individual labeling, and FDA also has a public list of all the products with post-marketing commitments, some of which are approved under Subpart H, and some of these products make that list. Just as with peptides, the key safety concern arise from differences to the reference listed drug. But the question is, does the immune response to these, is that warranted to be raised to the same level of concern as with peptides? We heard yesterday from Rob, from our friends at Eli Lilly, that no, there's no concern at all for oligonucleotides, right? That may be one way to look at it and part of the spectrum, but then others might say, okay, well, we don't know what the risk is, so we want to look a little closer and see if there is a real risk. I think, other than the immunogenicity risk, there's other effects of these oligonucleotides that may have other concerns that lead to safety or toxicity risk. There's potential for off-target effects, failure to target because of the nature of the product or the API impurity, differences in the delivery of the product, including the device constituent could result in differences. Unknown long-term effects, these sometimes may be difficult to ascertain due to inter-product switching, meaning that patients may switch from one product to another, and you really can't figure out what the problem is behind these. And this happens both in the peptide environment. For example, people will switch from semaglutide to tirzepatide to liraglutide or around based on whatever the endocrinologist is prescribing to them or what's available from the insurance company. So these things can happen, and sometimes it's difficult to discern. Was this long-term effect for immunogenicity resulting from specifically the use of your product? This has happened, we've seen in the biologics for psoriasis, for example, and there's lots of studies with products like Humira or Enbrel and looking at, hey, what's the long-term immunogenicity effect for these products? And it's complicated because of product switching. There are practical challenges for generics, API characterization, obtaining reference drugs. These are expensive. Imagine a product that costs, well, we know, we don't have to imagine it, $100,000 for a set of doses that you can figure out how to match with. And if you have to repeat that a few times, that gets pretty expensive to study. The immunogenicity concerns we mentioned, delivery and stability, and manufacturing scalability. These apply to a lot of generic products, not just for the oligonucleotides, which we're focusing today. Drug product sameness and impurities, the differences can result from API synthesis. All oligonucleotide drugs are synthetic by nature, which is a good thing, because then we avoid this whole issue of the cellular contaminants from recombinants. None of these are recombinant products, right? So you can avoid this whole conversation we had this morning in reference to oligonucleotides. Chirality and diastereomers, this happens, and especially in the nature of this synthetic scheme and what you're putting into the system to try to preserve the stability of these products. And the body is rife with RNases that are just angling to take out these oligonucleotides. API purification, we know that it's an issue, and can you purify out your problems, and the reference products are constantly finding better ways to purify. But of note, the reference products were allowed on the marketplace with certain impurities, and so if they continue to take out impurities that they think, based on what they're learning as they market, generics perhaps should be allowed to do the same. Something to think about. Excipient impurities. We talked about the excipients being concerned. If you have not just excipient impurities, but excipient differences potentially could result in some concerns, but that's something we can look at and discuss. Degradation in process versus storage. Both of these are something of concern. And during the manufacturing process, you may observe degradation of some of the sequences, some oligonucleotides, and then what comes out because of storage. And these are different storage conditions, shipping conditions, et cetera, could lead to some differences. So generic drug development, generic drug statutes and regulations apply to products that aim to be copies of NDA oligonucleotide drugs. We at FDA are committed to providing a clear pathway for generic drugs to ensure access to quality drugs for the American public. Some of the murkiness in our regulatory environment is just because we have some murkiness in the scientific conditions underlying what we're trying to do in the generic space. And so as we learn and we become clearer, we hope that the pathway becomes clearer. There's currently no ICH or FDA guidelines that specifically address the quality aspect and expectations for oligonucleotide drugs. So room to work on there. To assist generic drug development, FDA develops product-specific guidances for each reference listed product, including many complex drug products such as oligonucleotide drugs. I'm proud to say in our division, we work tirelessly to try to get PSGs out, and we've developed 13 PSGs so far towards oligonucleotide drug products, with more on the way. Right, Deyi and Eric? Yeah, Deyi's smiling. So we promise you, the American public, that we aim to get more of these guidances as new products appear in the marketplace. We also offer routes for direct advice in developing an ANDA submission, including the control correspondence mechanism, as you all know, for specific questions related to quality of bioequivalence, and the pre-development pre-ANDA meetings to propose alternative pathways from what's discussed in the PSG, or if there's not a PSG, you can propose a meeting and we will grant you that meeting, provided you have enough information to support a meeting that you submit. There are future tools. We have known unknowns. For example, what is the impact of impurities, or the variety of impurities and safety? There are possibly unknown unknowns, but we'll let you know what those are once we know them. That's a joke. No one's laughing. Okay. Tool development needs. We have advanced analytical methods, sensitivity assays, and immunogenicity models. We can see that these can continue to be advanced, and industry and agency, we're investing into these so we can learn more. A lot of it is targeting what we see as the highest risk, and right now we can see that the understanding of risk for this is somewhat divided. We heard from Rob yesterday from Eli Lilly. This is an NDA company, right? A new drug company that says risk is not there. So other folks can weigh in and say, okay, well, what do we think is the risk, and what is the relative risk of this versus other things that we want to entail and look at? So thank you very much. Appreciate your audienceship. Thank you.
Okay, thank you, Dr. Luke, for the very nice overview. Most people working in the oligonucleotide area probably know that the impurity profile for oligonucleotide drug are much more complex than peptide. So we will start the scientific discussion starting from the impurity characterization. So that's our second speaker, Dr. Kui Yang. The title of her talk is Comprehensive Impurity Characterization and Profiling in Synthetic Oligonucleotides Through Precision Analytics. Dr. Yang.
Okay. Hi, everyone. I'm Kui Yang. I'm a senior research scientist in the Office of Pharmaceutical Quality Research. My office located in St. Louis, Missouri, and I'm glad to see you all there. I'm presenting this from my office in St. Louis. It's my great pleasure to have this opportunity to talk about with you comprehensive impurity characterization and the profiling in synthetic oligonucleotides through precision analytics. This presentation reflects only the review of the author and does not endorse any commercial products or manufacturers. By this slide, I'd like to share with you our vision of pharmaceutical quality. Pharmaceutical quality is essential to providing American public quality medicine and assure our patients the safety and efficacy of every dose they are taking, as well as their next doses. Today, I'm going to elaborate on precision analytics for synthetic oligonucleotides from two aspects: characterization and impurity profiling. The analytical platform developed for this purpose is multi-attribute method for oligonucleotides, MEMO, which is LC, high-resolution mass spectrometry based and intended to assess identity, purity, impurities, and assay by a single platform. So first, characterization. How is this platform used in oligonucleotide characterization? Different from a conventional LC-UV method that may solely rely on retention time and comparison to reference standard, an LC-MS method can add another level of confidence by incorporating MS. However, to a different extent, depending on the mass spectrometry employed, for example, to a moderate level of confidence by pairing with a low or unit resolution mass spectrometry that measures average masses. Next, to a relatively high level of confidence by coupling with high resolution mass spectrometry, HRMS, that enables high resolving power and high mass accuracy. And lastly, to substantially high confidence by incorporating MS-MS-based sequence conformation. So built on HRMS, the MAMO platform can achieve high confidence in identity and characterization by utilizing the accurate intact mass measured through deconvolution of multiple charge states detected, or directly utilizing the accurate mass and isotopic peak distribution of individual charge states without relying on deconvolution algorithm. And additionally, with the feasible MS-MS capability, this platform can also add the sequence conformation by mapping the sequence using sequence indicating fragments. Those fragments are formed during tandem MS by cleaving the internucleotide linkages as shown here. However, to target a 100% sequence coverage, it requires sufficient fragmentation efficiency, which can be challenging in some cases. For example, samidine-rich and phosphorylate modified sequences may be difficult to fragment at individual linkages as shown in one of our recent publications. To overcome this difficulty, we have investigated and optimize the factors that can profoundly impact the fragmentation efficiency to maximize the sequence coverage. So next I'll move on to impurity profiling. Oligonucleotide products like peptide products we discussed the past day contain complex impurities that are often not only diverse in structure, but also with a wide range of abundance. To accurately quantify and profile impurities, it requires the analytical methods to be validated, which also assures their suitability in QC environment. We conducted a full analytical method validation following ICH-Q2R2 and ICH-M10 that evaluates, to just name a few, like specificity, linearity and range, precision and accuracy, and method sensitivities presented by the lower limit of quantitation LQ, etc. So given that impurities of synthetic oligonucleotides are typically complex, containing product-related impurities that are often structurally similar to each other, it is not surprising that underneath each individual peak, separated by the LC dimension, may often contain multiple co-eluting impurities instead of a single impurity as shown here for N-1 impurities. Among the co-elutes, some of them may be differentiated by MS dimension due to their mass differences. However, there are instances where the co-elutes have either identical or similar masses, making them inseparable by either LC or MS dimension. So for the remainder of my presentation, I will share with you some recent research conducted to specifically address this challenge. So what to do with separation by both LC and MS fails. Case study number 1, I will use the co-eluting isobaric impurity ions. So for example, N-1 impurities resulting from loss of uracil or cytosine nucleotides. When multiple charged, they typically form isobaric ions of identical nominal masses because their mass difference is only at 1 Dalton. We recently published our work in addressing this analytical challenge where we developed a HR-MS-based approach to overcome the limitations of a conventional LC-MS method in quantifying co-eluting isobaric ions individually. This approach has a simple and straightforward workflow as shown here. It requires 3 isotopic peak distributions measured by HRMS for both impurity standards and the sample. Once the 3 isotopic distributions are input, the composition of N-U and N-C in the sample is quantified by the automated simulation. Furthermore, when impurity standards are not available, the workflow then uses their theoretical calculated isotopic peak distributions instead, which offers a great flexibility for scenarios where reference impurity standards are challenging to obtain. And this figure showcases the quantification of individual U and C deletion or addition impurities using either the measured isotopic distribution of impurity standards as shown by blue bars, or their theoretical calculated isotopic distributions as shown by the purple bars. This method then allows one step further in in-depth impurity profiling than a conventional LC-MS method that generally reports only a total without differentiating individual isotopic components. And now I'm moving to case study number 2, co-eluting isomeric impurities, for example, N-1 impurities, where a same nucleotide is deleted from the full-length sequence, but is from different sites. For example, for a full-length sequence shown in this table, this is the same sequence as leucinosin. We can see there are 2 possible positional isomers for N-G impurity and 5 for N-U impurity. So a question that is fair to ask is whether the deletion site of a nucleotide from the full-length sequence matters. If the answer were no, then there would be no need to differentiate those positional isomers or quantify them individually. To gain some insight to this question, our collaborators at FDA and CTR evaluated the toxicity of selected N-1 impurity positional isomers using primary human hepatocytes and compared with the full-length parent sequence. As shown in this figure, some impurities shows similar toxicity to the parent sequence. Some were more toxic than others. For instance, the N-U positional isomer 3. And currently, the immunogenicity risk assessment of those impurities is ongoing within CDER, led by OPQR Dr. Daniela's group. We hope to encourage more thorough risk assessment of those structurally similar impurities, and hopefully from this audience as well. Those assessments can help answer many questions such as to which level the impurities should be characterized and reported. For instance, to N-1 impurities as a group is adequate, or one step further to individual N-1 impurities, or even further to discrete positional isomers is required for product safety and quality control. From analytical perspective, we're asking a different question, that is, for which level of impurity characterization the current analytical capability allows. In this scenario, N-G impurity has 2 possible positional isomers, although by MS dimension, they are non-differentiable. By tandem MS, notably, diagnostic fragments to each individual isomer can form from the backbone cleavages within the yellow highlighted blocks, where the deleted G would reside in the full-length sequence. Those unique fragments identified can then be used for identify and quantify the positional isomers. So in this more complex scenario, N-U impurities have 5 possible positional isomers. By tandem MS, it may be challenging to resolve them all because many fragments would be shared among those 5 isomers. Therefore, defining unique fragments to each isomer would be impossible. I-mobility is a newer technology that adds an additional separation dimension based on molecular shape and mobility, orthogonal to LC and MS dimension. Our preliminary study on application of I-mobility on N-U isomer mixture and this recently published paper on separation of oligoisomer impurities using high-resolution I-mobility indicates that I-mobility is promising to provide the separation of complex mixtures where LC and MS may not be sufficient to separate them. So to conclude, here is a revisit of the impurity complexity in synthetic oligonucleotides, arising from the diversity of product-related impurities that are often structurally similar to each other or to the parent sequence. Common impurities include the deletion or addition sequences, as well as unintended modifications at backbone or nucleotide units, each of which as an impurity family may be separable by chromatography from each other, such as N-1, N+1 impurity families. Delving deeper, depending on which nucleotides are being affected, each individual deletion or addition impurity family may comprise multiple members that, although likely co-eluting, may most likely be differentiated by MS dimension because their mass may be different for most of the time. Furthermore, depending on which site of a nucleotide on the full sequence are being affected, each impurity member may be composed of multiple components that not only co-elute but also have identical masses that's unresolvable by either LC or MS dimension, which poses the greatest analytical challenge in their separation, characterization, and quantification. This is why and where we have been devoting our efforts on. The 2 case studies I share with you today may just represent a tip of the iceberg with a lot more remaining unresolved. On the other hand, risk assessment of individual impurities, including their immunogenicity and the toxicity test, would serve as excellent supporting evidence to help address the question whether or to which level impurity characterization and profiling should go to ensure product quality and safety, which may be more profound in conducting impurity comparative studies for generic products. So in summary, today I have discussed with you a fit-for-purpose analytical platform, which is a LC HRMS-based multi-attribute method designed to facilitate identity, assay, purity, and impurity analysis of synthetic oligonucleotides using a single analytical platform. We have validated the central LC HRMS following regulatory guidances. We have also dedicated significant efforts to in-depth impurity profiling by utilizing high-resolution mass spectrometry and emerging technologies such as high-resolution ion mobility, particularly focusing on resolving challenging impurities, for instance, those co-eluting isobaric or isomeric impurities, otherwise most likely not resolvable by conventional LC or MS dimension. And lastly, I would like to take this opportunity to acknowledge the generous support from multiple offices of FDA, particularly Office of Pharmaceutical Quality Research, both Analytical Division in St. Louis and the Immunogenicity Group, Office of Generic Drugs, who supports our ORAS fellows in regulatory science research, and FDA and CTR, who collaborate with us for impurity toxicity assessment and the complex product PST development oligo SME triage team. And we are also very honored to receive multiple FDA research grants from Critical Path and the Chief Scientist Challenge programs that significantly facilitate oligo research at FDA. And I would like to thank you for your attention. I will come back for the panel discussion for your questions. And thank you.
Okay, thanks, Dr. Yang, for your research and a wonderful presentation. Next, we move to our third speaker, Dr. Bart Rogers. He's also going to give us an online presentation. The title of his talk is Regulatory Oversight of the Immunogenicity Risks of Oligonucleotide Therapeutics. Bart, go ahead.
Okay, thank you. Hello, and first off, my apologies. I wish I could be there today. I'm actually out of the country attending another oligonucleotide talk, so again, my apologies. Today, I was asked to discuss the regulatory oversight of the immunogenicity risk for oligonucleotide therapeutics from a regulatory perspective when we do at the FDA. For those of you who do not know me, I'm a pharmacologist in the Office of Clinical Pharmacology in the Division of Translational and Precision Medicine here at the FDA. So what exactly are oligonucleotides? These are modified nucleic acids that are typically designed to bind to a complementary RNA sequence and alter the subsequent downstream protein. These effects are typically mediated through Watson-Crick base pairing. Now, these oligonucleotides have been synthetically modified to increase specificity, slow degradation, and specifically to avoid immune recognition, as I'll get into in this talk. Now, there are various means of this synthetic modification. There are backbone alterations, for instance, phosphorothioate or a 6-membered morpholino ring or common substitutions there. Another common position for substitutions is at the 2' position of the ribose sugar, where oftentimes we'll see fluorinated substitutions, monoethoxyethyl, constrained ethyl, just to name a few. Now, if you'll notice in the bottom left-hand corner, this is a single strand depicting a single-stranded antisense oligonucleotide, a very common chemical moiety in this field. This is known as a 5-10-5 gapmer, where on the 5' and 3' ends, we have RNA oligonucleotide bases, and then the gap, we have 2' deoxyribo bases. And this is to support an endogenous endonuclease known as RNase H, which then cleaves the messenger RNA sequence that the single-strand antisense oligonucleotide binds to. Now, I'll draw your attention to the bottom right-hand side. This is the other most common moiety in the field, which is the double-stranded siRNA. Here it depicts with a GalNAc conjugation to facilitate hepatocyte delivery. This double-stranded oligonucleotide works using a different endogenous RNA nuclease known as Argonaute-2, which is part of the RNA-induced silencing complex. More importantly than what exactly are RNA oligonucleotides is the why, and this area really has me excited. If you'll notice a central dogma, DNA, RNA, and protein in terms of translation, transcription, translation, and direction of molecular data. So for the most part, small molecules and biologics have really only interacted at the level of the protein. Whereas now with RNA oligonucleotides, we can target many things upstream, working at the level of the RNA to silence aberrant proteins or increase expression of needed proteins. More importantly, we are now able to target the cause of many genetic disorders that were previously considered undruggable with small molecules and biologics. And again, these utilize endogenous cellular machinery. And as I mentioned, they can increase expression or an agonist-like effect, if you will, or silence expression of pathogenic proteins for an antagonist-like effect. Now, on the last slide, I showed you both antisense and single-stranded antisense and double-stranded siRNAs. Once a chemical moiety has been approved and you understand that, they're able to be quickly synthesized. To date, as Dr. Markham mentioned, the FDA has approved 17 RNA oligonucleotide products. The first of these Viversen back in 1998. This was an intravitreal injection for CMV retinitis. It is no longer marketed, all the way up until imetelstat in 2024, with many approvals likely to come in the future due to the increased exposure this field is getting and the increased development that's being observed in the IND space. A couple things I want to note here. Of these 17 approvals, 11 of these have been for single-stranded antisense oligonucleotides, and 6 of these have been for double-stranded siRNAs, as noted by the siRAN suffix here. A number of these, 2 of these are no longer marketed, and a number of these have been approved under conditional approval pending confirmatory evidence in the post-marketing space. When evaluating safety of these compounds, we look at 3 things: on-target safety, which is beyond the scope of today's talk, and then off-target safety. In off-target safety, I typically look at 2 things: hybridization-dependent safety, as well as hybridization-independent, which I'll get into in a moment. So hybridization-dependent safety, off-target safety, is what it sounds like. It's mediated through Watson-Crick hybridization. However, you're binding to a messenger RNA that is not the targeted messenger RNA. It's important to note that this is not always detectable in animal models if the target is not present. Some targets are not conserved through evolution, especially not in the rodent models, so in which cases we'll have to use a surrogate oligonucleotide to assess the off-target hybridization effects. However, there are cases where you can use knock-in mouse models as well. Typically, off-target hybridization is best assessed through various bioinformatic approaches, such as a blast search or a bow tie algorithm, to assess the potential off-targets that the oligonucleotide may represent. Now, when evaluating off-target non-hybridization dependent, this is typically mediated through the innate immune system. Now, our innate immune system is pretty good at recognizing foreign RNA and DNA, as these have been evolutionary threats to us throughout evolution, throughout human evolution. So there are a number of immune pattern sensing receptors that the RNA oligonucleotides must avoid. So for instance, you have toll-like receptors, TLR receptors that bind to certain single-stranded RNAs, single-stranded DNAs, likewise, RIG-I pathways, STING pathways, and many of these are very inflammatory pathways where you'll see downstream interferon activation as well as other inflammatory mediators. And it's important that oligonucleotides avoid these, otherwise they're not going to be very well tolerated. Many patients do not like experiencing these effects. Again, there is still some questions about the dose exposure dependency here, but again, most oligonucleotides are selectively modified, synthetically modified, to avoid these pattern sensing receptors. Now, as part of this, a number of industry individuals and FDA folks put together an oligonucleotide safety working group, and we published in 2022 an assessment of the immunogenicity potential of oligonucleotide-based drugs, and I'll just provide a brief overview of this paper here. So it's important, first off, to denote that oligonucleotide therapeutics represent a diverse chemical subclass with various routes of delivery, so many things that can influence the immunogenicity potential of these. It's very important from recommendations from this white paper that sponsors conduct an immunogenicity risk assessment early in product development and then follow a risk-based approach based on the product's specific features, for instance, backbone modification and patient-specific factors of the population that you're looking at. It's very important to evaluate the potential to interact with these pattern recognition receptors that I was just mentioning, things like toll-like receptors, And currently, neutralizing anti-drug antibody assays are not part of regular assessment for oligonucleotide therapeutics. Now, if you look to the decision tree on the right here, again, it's very important early on to determine the immunogenicity risk potential. Many oligonucleotide therapeutics are thought to be low immunogenicity risk. However, this can certainly change with alterations to chemistries and backbones. If in the case where the risk category is thought to be high, we recommend, FDA recommend, or the OSWG recommends a collect and analyze strategy. Whereas if the immunogenicity risk is thought to be low, the FDA recommends a collect and hold strategy. And I'll go into a little bit more detail in some of these aspects regarding the FDA's recommendations. So as part of this, the FDA issued the finalized clinical pharmacology considerations for development oligonucleotide therapeutics. Immunogenicity assessment was a key factor in this guidance. And the FDA, along with OSWG, recognize that it's very important for sponsors to develop an anti-drug antibody assessment prior to submitting their testing samples from their pivotal studies. It's very important that sponsors validate the assay and, again, submit these reports to the FDA. Now, if compounds are agreed to be low risk, immunogenicity assessment may be deferred to the later stages of development. Now, in the case where oligonucleotides are thought to be low risk and the chemistry supported by prior approvals, a collect and hold testing strategy may be justified. Again, not in all cases. We recommend sponsors do reach out to the FDA. When looking at our current database of immunogenicity with the current approved 17 oligonucleotides, I'll present in the following 2 slides. There's a couple things I want to quickly draw your attention to. First off is the second column here, the route of administration. Again, there's various routes of administration presented here. And then importantly, the third and fourth columns, the anti-drug antibodies, yes or no, and the percentage of anti-drug antibodies. While our eyes are drawn to this, it's important to recognize that the anti-drug antibody percentages are not comparable across drugs, given the specificities with each of the assays involved in anti-drug antibody assessment. So again, I want to point that out to you as our eye sort of likes to compare things. These numbers are not comparable. They're very dependent on the specificity of the assay. And then last, I want to point to our current labeling language for these anti-drug antibodies. So we'll start off with mepomarsen, approved in 2013. Here was a unique case where we actually did see, and it's the only case where we saw flu-like symptoms were higher in anti-drug antibody positive patients, and anti-drug antibody positivity did lead to increased C trough. With eteplirsen, the next approval in 2016, anti-drug antibodies were not measured. In 2016, nusinersen was approved. Now, I should note here, this is an intrathecal oligonucleotide. However, despite intrathecal delivery, the oligonucleotides rapidly cross out of the blood-brain barrier into the systemic circulation. Here, the anti-drug antibody percentage in the pivotal study was 6%. But I do want to note here that 5 of these patients did have transit anti-drug antibodies, which points that the innate immune system did see the anti-drug antibody and did not decide to see it as a threat and mount a response over repeated dosing. If you'll note here, with many of these compounds in labeling, there was no effect on the anti-drug antibodies on clinical efficacy, safety, or PK. I don't want to. Looking at the remainder of the compounds here, just a couple things I want to make a distinction of. Tofersen, another intrathecal intrathecal oligonucleotide, did have 58.4% positivity of anti-drug antibodies, and they did appear to decrease plasma clearance by 32%. However, the effects of anti-drug antibodies on plasma clearance are unknown. Again, this drug acts centrally within the CNS, and there was no effects on safety. Just this year, the approval of eplontersen, there were a presence of anti-drug antibodies that did not affect plasma Cmax or AUC, but again, they did impact C trough. But again, overall, the majority of these compounds have demonstrated no effect on clinical efficacy or safety of PK, despite having some anti-drug antibody positivity. Looking in summary again of our current database, there have been no clear impacts on efficacy or safety, and there are some limited impacts on the pharmacokinetics of these compounds where there were anti-drug antibody positivity, specifically in increased C trough. Development of anti-drug antibody tends to occur after multiple doses, and it is transient some. there is some learnings there that, from single dose studies, what can be gleaned. And the optimal timing for sampling may depend on the chemical class of the oligonucleotide and the specific chemistry. When assessing immunogenicity, the route of administration may certainly alter immunogenicity, and FDA is aware of this. Again, to underscore the importance, the assay sensitivity and specificity may certainly impact the interpretability of these anti-drug antibody findings and effects on PK, efficacy, and safety. Importantly, oligonucleotides that have an intended immunomodulation as their mechanism of action, for instance, CpG receptors, may need to be evaluated in a different manner. For instance, animal studies may provide further information regarding intended and unintended effects. Again, there have been no approvals with CpG modulatory oligonucleotides to date, but again, there are some of those in development. Some important considerations: single-dose early phase studies of limited duration are not designed to elicit anti-drug antibodies, so they may not be valuable for assessing that. As I mentioned earlier, a collect and hold versus collect and test strategy is something to consider based on the overall immunogenicity risk profile of the oligonucleotide. Early assay development and validation is critical, and to date, neutralizing assays have been challenging to develop and are not typically required at this time. When relating to today's talk and the purposes of this conference, concerns over impurities in the generic space for immunogenicity, it is important to note that impurities include single nucleotide-based variations that may be immunogenic, and assay and development and validation will be of critical importance when assessing this in the generic space. With that being said, there are a number of folks at the FDA that I would like to acknowledge for assistance with these slides, as well as assistance with our clinical pharmacology guidance, and they are listed here. And I will stop there and take questions at the end during the panel discussion. Thank you.
Thanks, Dr. Rogers, for the very nice overview and also sharing the regulatory oversight on those approved oligonucleotide drugs. Next, we will move back to in-house speaker, and I want to invite Dr. Sudhir Agrawal to give us a talk on oligonucleotide structure and also innate immune activation.
Thank you, Deyi. Thank you, Daniela, for the invitation to speak. It's my pleasure to share the background on oligonucleotide therapeutics. So as I think Bart gave a very good introduction, so what we're talking about is use of an oligonucleotide as a drug, and it could have multiple mechanisms of action. So the oligonucleotides are designed to bind to mRNA. It could be coding RNA or non-coding RNA. And depending on the sequence and chemical modification, it could recruit multiple mechanisms. It could be simply antisense. It could be splice modulation. It could be siRNA, or it could be guide RNA. In all cases, the first step is to oligonucleotide to bind to the target. The second step will be engage machinery, and the ultimate goal is to modulate the translation of the protein. But at the same time, the oligonucleotide we are using could be recognized as a pathogen-associated molecular pattern, which could result into an immune response, which would be an unintended mechanism. And that's the balancing act between choosing the right sequence and the right chemistry. So what I'm going to do is really divide the talk in three portions, really take you back into history when we started to see these observations, which we had no idea about the immune system, and then come back to really talk about what we know about the innate immune system and how the chemistry affects the immune activation. And then in the third portion is what we have learned, how can we use it to create a better therapeutics going forward. So this goes back to 1987, very early days, and we wanted to use oligonucleotide as therapeutics. And the first step was to provide the stability. In this case, the backbones were modified. So in those years, we modified both RNA and DNA. So what you see in sulfur is S completely replaced to oxygen, and that provides the stability. By replacing sulfur, now we are creating a diastereoisomers. There are two isomers. Generally, they are in 1-to-1 ratio. But if you're thinking about a 20-mer long DNA, you have a 2 to the power 19 isomers. So they are not purified. As we progressed the field, we learned that for DNA phosphorothioate, which is the first structure, activates RNase H, means when it binds to the target, it activates endogenous enzyme called RNase H, and that degrades the RNA. So this became the preferred choice of first generation antisense, and about 20 plus candidates went into clinical evaluation using this chemistry. They were for antiviral to anti-cancer agents. They were from phase 1, 2, and 3 based on preclinical studies, based on animal model studies. But most of these candidates have been now discontinued. The reason for that was we started to learn that these class of compounds have immune responses or polyanionic characteristics because we have stabilized them, so they're bringing these unwanted side effects. And then the question was also whether the mechanism of action is intended based on the target or is it coming from the immune activation. So from that point of view, I'll share with you two observations. One was in 92, 93 when we were studying these compounds first time in primates. We had seen serious hemodynamic changes and we had lost a couple of monkeys. And the field was completely at standstill, not knowing what is happening here. And it took us about 18 months to solve it, and it was related to complement activation. And it was related to rapid infusion of the compound, because that compound was charged material. So it drops in blood pressure, drops in complement, and it also activates C5a, and that remains true to date. But over thousands of patients have been now dosed, but in all of these cases, it's a slow infusion. So it's a dose-dependent, rate of infusion dependent, and it's chemistry dependent. At the same time, we also saw this leads to thrombocytopenia and neutropenia as well. It was transient, again, depending on the rate of administration dependent and dose. It also affected the clotting factors, prolongs the APTT. So after about 2 years of work, it was established that this is a chemistry-related, not sequence-related, but it was applicable to all antisense compounds and dose-dependent. So since then, all the compounds have been given by slow infusion or subcutaneous to avoid the maximum plasma concentration. So this was in primates, not seen in the rodent animals in earlier studies. And the other observation we made was when we went into clinical studies with the same compound, that the patients who received intravenous infusion had complained of flu-like symptoms and chills and induration at the site of injection. This is again mid-90s. And we collected the blood sample from these patients, but in those cases, we could not analyze the samples for HIV levels. When the samples were analyzed, then we found out we were suppressing the virus rather than we were activating the virus. So this was leading to increase in viral load in blood sample. The trial was discontinued, but the observation was made when a compound is given by subcutaneous route or intravenous route, there was an induration and flu-like symptoms. There were many theories at that time that it could be an endotoxin present in the manufacturing batch or is a sequence related. But as we wrote much later after collecting a lot of data, that this could have been an immune activation through a CpG mediated immune activation through toll-like receptor 9. So this is not local, it's systemic. It leads to flu-like symptoms, but it also leads to retrovirus activation as well. So again, as I said, these are all the compounds in those years were mixtures. So there was a theory that it could be stereoisomer dependent. So we made both R and S isomer, separated it out. And it came out that R isomer is not stable enough, but it does cause increased immune activation. But S isomer is more stable. It causes more polyanionic related side effects. So the idea was not to pursue the separation, at least for in a longer oligo. But certainly recently there have been reports of specific position changes for stereoisomers. So First 10 years were development of antisense as a first-generation phosphorythide DNA. At the same time, as we started to see these characteristics are going to limit the development of these class of compounds, we continue to develop RNA modification. RNAs were binding to the target, but not creating the degradation. But then they were less potent, but then we went back to analyzing whether to see if these immunological phenomena and polyanionic phenomena are different than in RNA. And to our surprise, complement activation or clotting factors or immune activation were completely mitigated compared to DNA. So that is not the charge dependent of the phosphorythide in DNA, but rather a DNA dependent and not RNA dependent. And so when we had this data, it all came up is that how can we bring the desirable properties of these two modifications into one so we can modulate it? And that's what led us to create what we call now gapmer chemistry. The top one is a mixture of a RNA and DNA into one. So RNAs are placed at the end, DNA is placed in the middle, and the bottom one is completely RNA for splice modulation. So these two chemistries were introduced in mid-90s, and now the foundation for all the drugs which are being approved based on antisense platform. So again, drugs are being approved, but there are the number of drugs which are also failing at different stages could be for many reasons. It could be the failure of translation of biology. But as you read more about it, it is really off-target inflammatory targets, inflammation or safety signal. So are we still seeing the inflammation coming from the sequences and the chemistry, and how shall we analyze those going forward? So in mid-90s was a very confusing period because we were seeing this immune activation we had no guidance on because the receptors were not even identified. So we are only playing with the sequence. So if you look at the first figure, we truncated the sequence. We were thinking that the longer the sequence might be causing it, so we truncated the sequence. The first time we truncated sequence from the 5' end, we saw loss of immune activation. But when truncated the sequence from the 3' end, we did not see loss of immune activation. So it's not length dependent. But I highlighted that in one case, we were deleting the CG. And that was avoiding the immune activation. But at the same time, we were also working at this, what we can call gapmer chemistry. And in the gapmer chemistry, whenever we modified the CG, we were also mitigating the immune activation. So there were guidance that certainly is the sequence, but also chemistry can modify them, and we can mitigate the immune activation. So this was really in dark. We were playing and learning about it. This is we published in mid-90s. But at the same time now, when we look back, our purpose was to create an antisense which binds to RNA target. But now we know that the protein binding leads to, due to polyanionic nature, leads to complement activation, and that's a dose and infusion dependent, and that can be avoided. But the immune activation is sequence and chemistry dependent, and how can we learn more about it based on the current knowledge we have? And so that takes me to the second topic of the talk, which is oligonucleotide-mediated immune activation. So in this case, again, mid-90s to for 10 years or so, the field was very gray. There were no clear indications. There were reports coming out from NIH that the CpG-mediated immune activation, there was CpG DNA was being used as vaccine adjuvants, and in 2000, finally, the paper came out that toll-like receptor 9 recognizes bacterial DNA, which is unmethylated. In parallel, we were working on understanding how these compounds activate immune responses, what are the toxicological effects of these compounds, which have a different backbones and different chemistries, and then also these compounds, which are immune stimulatory, could have an anti-cancer and antiviral activity because of the immune activation, so there could be a pan-antiviral agents. But then the picture became clear, and again, Bart gave a very good introduction behind these receptors. So now we know there are pattern receptors which recognize different pathogen-associated molecular patterns of nucleic acid. They are both present in endosome and also cytosolic. Depending on the nucleic acid of nature, they could trigger a pathway. which will lead to cytokine induction. These are expressed differently in mice and in primates and humans, so that complicates how we assess them because the preference is different depending on where they are expressed. But each receptor has a different recognition pattern, some for DNA, some for RNA, some for single-stranded, some for double-stranded. And some have 5' capping agents, some have different modification, backbone modification. And that all leads to engagement of the receptor and then following cytokine induction. They are expressed on different cell type. And that also becomes very important how to assess them. A mixture of cell population might be an ideal way of looking at it. Purifying the cells might not lead to, and missing out the cell population which is expressing the target. So what we have done is I'm going to focus only on TLR9 because that is for bacterial DNA and that's more relevant for antisense. But there are portions of RNA in antisense as well. And for that, we need to focus on TLR7, 8, and 3 as well. But am I going to focus only on TLR9 today? So given a sequence of any DNA sequence which has a unmethylated CpG motif that will be recognized by TLR receptor 9 and will activate the process, depending on where the CG is in the sequence from 5' to 3', the nature of immune activation will vary. It's not that presence of CG will lead to immune activation. It depends on where it is. If it is toward the 3' end, it might not be activated at all because it does require a following sequence from the 3' end. So what we have done is really systematic studied where it is, what modification lead to suppressive activity, what modification lead to increased activity, can it be substituted completely with modified bases? And for that, we published a library of modifications which could lead to engagement of the toll-like receptor 9 or completely mitigating it. So two properties here. If we modify the 5' end, we can completely mitigate the immune stimulatory process. So 5' end is required for immune activation. So if you keep that in mind, we'll come back to how can we use that knowledge in creating better therapeutics. So a couple of examples for it. So there are two examples here. One is listing the sequences. You see in the blue is the CG is in the middle. But the modifications are being introduced away from the CG. First with the RNA modification, which is routinely used in antisense and other nucleic acid therapies as well, 2' substitution. So if you place it at different positions from 5' to 3' end, immune activation completely varies. So close to 5' end, 2, 3, 4 nucleotides away, that modification neutralizes CpG-mediated immune responses, but not the others. But in some cases, it increases the immune activation. So it's not the sequence alone, it's where the modifications are. Similarly, another example in which the charge is replaced with one modification, which is called methyl group. And in that case, very similar observation, 5' end does lead to modification and mitigation of the immune activation. Similarly, if we take another example in which the sequence has two CGs. So if you look at in this case, there are, So in this case, there are two CGs placed at two distant sites, and that leads to NF-κB activation and leads to cytokine induction in mice. If you modify one CG towards the 5' end with the GA, and GA is RNA modification, sequence is not changing, we're just changing the backbone. NF-κB is mitigated, even though the compound has a CG present. So it means the 5' modification has now captured the toll-like receptor 9. Second CG presence is not needed. But if you modify the 3' end CG, the 5' CG is still recognized and it activates the immune system. But if you modify both CGs, now we have completely mitigated immune activation. So again, it's the importance of sequence and chemical modification, which leads to immune activation in both in cell culture and in vivo. It has no activity. So the one I just showed you, the compound which has no activity, it doesn't mean it's inactive. It's not activating the immune system. Rather than it's bound to toll-like receptor 9 and occupying it, so toll-like receptor 9 is non-functional anymore. So it's really acting as an antagonist. So now if you take a toll-like receptor 9 ligand and test it, it blocks the toll-like receptor 9 activation. So that could lead to immunosuppressive characteristic of the compound. And that needs to be assessed because it could have safety signal based on immune suppression by a nucleic acid, not only activation. So again, interestingly in this field, immune activation portion of or characteristic of the compounds are being exploited as therapy, as an immunotherapy for a variety of diseases. It's approved as vaccine adjuvant for hepatitis B, but many indications for others, clinical data exists, proof of concept exists, but not approved yet. And so these compounds are being used for immunotherapy of cancer. So these are side effects from antisense days, but now have become therapeutic approaches and are being used for different indications. So I'll take one example, translation of the compound I had worked with. This is a TLR9 agonist. It's a DNA, which is 2 11-mers linked together on the 3' end. So in mice in vivo data, primate in vivo data, and humans in vivo data, so it translates very well from preclinical to larger animals to human trials. You get the cytokine induction, and that also can be reproduced very well in human cells. And so these class of compounds do have a reproducibility from preclinical models to human trials. And there are well-reported assay systems to understand the characteristic of these compounds, except in human PBMCs, it's gender, age dependent. So this, again, if one is studying them, then you have to look at number of donors to assess the profile. So it comes back to, as we think about these class of compounds, so it's sequence dependent, and I talked about modification dependent. So it could be hybridization dependent or independent. So they are just causing the immune activation through a pattern recognition receptor or binding to a protein, which could be polyanionic related effects. And the immune activation through a pattern recognition receptor will lead to pro-inflammatory cytokines, will lead to anti-drug antibodies and thrombocytopenia. But in the other cases, if the compound continues to be built up in the tissues, because these compounds have a long half-life, they could lead to damage-associated molecular patterns intracellularly and may cause damage signals as well, and that will be inflammatory as well. So those can be assessed easily. Some of these can be assessed using cell culture model. So now we know that these compounds are binding to these receptors. We know they're binding to proteins. So are they consuming the active compound, which could be binding to mRNA? So they are serving as a reservoir to keep on absorbing these compounds and taking away the active compound. So how can we use, based on this knowledge, the chemical engineering to create better compounds? So in this case, antisense designed to bind to RNA, now we know that they are binding to proteins, they are binding to pathogen-associated molecular patterns, pattern recognition receptors, and they could be also binding to non-targeted RNA. So can we avoid these and maybe create a better compounds? So I'll take one example. In this case, the idea was, I told you that we can mitigate immune activation just by blocking 5' end. So you can imagine the dark circles are your antisense compound. We are just extending it by a short piece, which is a natural DNA. And this structure will be formed because the way the open circles are created, they are complementary to the other side of the DNA, and it forms a structure. It's the same molecules, one single molecule, doesn't have a 5' end, so it mitigates immune activation. Since it has a certain shape, it also avoids binding to proteins. It can get into the endosome and get out faster, and so it's present in cytoplasm. It's not opening until it finds the target. So the region which is in the dark circles, they only will bind to the target, and that will force it to open at the site of action. So with this, we can see some data. So if we take for a one target, PNP-PLA3, the top one is a gapmer, which is widely used in the industry. We created this in mid-90s. The other structure is called splitmer. It's another version of a gapmer. And then the two bottom ones are one which we just took the same sequences and created into what we can call cyclic structures. And these structures, when we study them in cell culture, with no other change made, we are seeing over 20-fold improvement in potency. So that means we are avoiding the immune activation, we are avoiding the protein binding, and by avoiding those, not only of mitigating the side effects, but we are taking that away and creating compounds which are more potent as well. So there are a number of cyclic structures and different modifications which are introduced in cyclic structures, which are now permitting us to create better oligonucleotide therapeutics. So with that, in summary, chemistry and sequence has helped. The gapmer and the modified RNA are being used in antisense. Immune activation is an important factor. It is sequence and chemistry dependent. But at the same time, the chemical engineering, I call it, not chemistry, just to take the same building blocks and put them in a different way, and is permitting us to create better compounds. And with that, I'm grateful to my colleagues over the last 30 years who have been worked with me on these things. And as you can see, in thinking about the next generation therapeutics, is the chemistry and also the innate immune responses have to come together in creating a better therapeutics. Thank you.
Yes, thank you, Dr. Agrawal, for a very insightful talk. Yesterday, we spent a lot of time, you probably know, discussing Hermes, and that's for peptide product. Today, we also have the same impurity, same type of impurity for oligonucleotide. So next, I want to invite Dr. Hannah Lee to talk about the assessment of Hermes for oligonucleotide therapeutics.
Thank you for the kind introduction, and also thanks to the organizing committee for inviting me to give a talk today. Yesterday's session provided a comprehensive overview of RMI assay, and today I'd like to focus a little bit more on RMI assay in oligonucleotide therapeutics. And this is just a slide from my office. This talk reflects my view and should not be used in place of FDA guidance or policies. And it'll include some recommendations based on the regulatory experience and current thinking, not laws. Okay, the risk of drug product can be determined by looking at both probability and consequence. In the case of generic products, API is same as the reference product, and population treatment is also same. So the most residual uncertainty is coming from impurities. And you all know that there are two types, product-related impurities and process-related impurities we concern, and both of them are known to impact on safety or efficacy. So for new drug products, clinical studies can be used to assess immunogenicity risk. However, for generic products, we do not require clinical studies. Instead, there are tons of non-clinical studies can assess the immunogenicity risk of the generic products. We know it's not sufficient to determine immunogenicity risk. However, it can support to assess the relative risk compared to the RLD. So product-related impurities, they are directly associated with anti-drug antibody production and responses. So we understand it's a risk assessment is really required, but why do we concern process-related impurities? Yesterday, we learned that these process-related impurities also can increase the immunogenicity risk. That's why we are calling them IRMI, innate immune modulating impurities. So let's look at a little bit of their function. So those IRMIs can increase the maturation and activation of antigen-presenting cells and can facilitate peptide loading on MHC molecule and also can alter epitope dominance. And when we inject the product by subcutaneous, the RMI, which is injected with the product, they can promote the migration of antigen-presenting cells from this subcutaneous space to the lymph node, and then that APCs can present what they have to the T cells and B cells in the lymph node. And RMI also I said they can induce the APC activation, and they are mostly produce pro-inflammatory cytokines such as IL-1, IL-6, IL-23, which lead to the transition from Th1 to Th17 responses. And also these impurities are known to activate B1 cells through the TLR-BCR crosstalk, and this can increase antibody production and class switching. So everything says that these IRMIs can change the quantity and quality of immune responses. For generic peptide products, so I'm talking about the peptides, the immunogenicity risk assessment is done by two parts. One is the product-related impurity, and the other part is process-related impurities. If you see any product-related impurities that are new or present higher compared to the RLD, and it should be more than 0.10%, orthogonal methods are used to assess MHC binding and T cell activation. So those are for individual product-related impurities. And it is also important to assess whether your product has any impurities, including product and process-related impurities. They can activate innate immune responses, and RMI assay is used for that purpose. Okay, these are generic peptides. Then what do we need to test for oligonucleotide therapeutics? Generic. There are some challenges to answer that question because, so far there are 19 FDA-approved oligonucleotide drugs, but they are relatively new and they are targeting very rare disease. So it means clinical experience is very limited. So that is one reason. And currently there's no FDA or IHC guidelines on immunogenicity risk assessment for generic oligo drugs. Draft product-specific guidance exists for 13 products, but all of them just say, consult the agency if you have any questions about the immunogenicity risk assessment. And as you learned from Dr. Yang's talk, there are another challenges coming from sheer number of impurities in this type of drug product. So it makes us impossible to assess the individual impurities immunogenicity risk assessment. But there are certain things we can consider. It's not like the all, just based on our experience and current knowledge. There are a few things we can consider for immunogenicity risk assessment. So in this type of drug, product-related impurities, we do not think they increase the immunogenicity risk through MHC binding or antigen-specific B or T cell activation. Instead, we are thinking both product and process-related impurities, they can induce local inflammation or they can have immune modulating effects. Actually, those are very problem for the drug, for the intrathecal injection products such as nusinersen, because local inflammation can pose a big significant risk. And RMI assay can be used to assess the immune modulating or inflammatory effects of impurities. So I'm going to talk more about that in the next slides. And Dr. Agrawal mentioned about these too well. Some clinical and non-clinical data show that oligonucleotide drug products pose a risk for complement activation and thrombocytopenia. So these should be also considered because impurities can increase those risks. Let's look at some nucleic acid sensors. In our body, there are several nucleic acid sensors can bind to double-stranded RNA, single-stranded RNA, and DNA. And these are, I just brought some examples. And these TLR3, RIGI, MDA5, LGP2, they are known as double-stranded RNA receptor. So potentially, in theory, siRNA therapeutics or the functional aptamers can bind these receptors. TLR7 and TLR8 is known as single-stranded RNA receptor. So in theory, as ASO aptamers and denatured siRNA, they can bind to these receptors. There are also DNA binding receptor, DNA aptamers and DNA therapeutics can bind TLR9, CCAS, AIM2. But as shown in this table, not all these double-stranded RNA or DNA, they can bind to these receptors. There are certain criteria, certain characteristics needed for the ligands. So for example, let's see TLR3. Double-stranded RNA, if they wanted to bind to TLR3, it should be longer than 21. But with the 21 base pair, it's not induced strong immune responses. To trigger strong immune responses, it should be more than 45 base pair. So when we design, to trigger the immune response through these receptors, the oligo should have certain chemistry, structure, length, and sequence. So when we design the oligonucleotide therapeutics, those possibilities should be considered. So indeed, it's considered. Yeah. So this shows that common modifications used in FDA-approved oligonucleotide drugs. So as Dr. Agrawal explained, a lot of oligo drugs has PS backbone to increase their stability, but unfortunately, this PS backbone can trigger TLR signaling. So to reduce these unnecessary immune responses, these oligo drugs has another modifications, such as second prime ribose substitution or nucleobase modifications. So second prime OH group is replaced with methyl MOE or fluoro groups. So it can reduce the immune responses. And also we know that methylated CpG unmethylated CpG can trigger TLR9 signaling. So we know about that. So reduce TLR9 immune activation. This base, if it's cysteine, it's replaced with methylcysteine. And also siRNA, there is 2 nucleotide overhang to prevent binding to the RIGI or LGP2, which recognize the blunt end RNA. So there are a lot of efforts to reduce unnecessary immune responses. But let's say, what if the generic drug manufacturing system increase the level of impurities lacking those modifications? What happens? It can increase immunogenicity risk. and also the process-related impurities, including organic, solvent, leachables, or other contaminants in drug product, that they can also induce innate or inflammatory responses. Then coming back to the oligonucleotides, can we detect RMI in the oligonucleotide therapeutics? For that, we need to develop assays that are fit for purpose. So yesterday, Dr. Thacker explained about these fit-for-purpose assays for the RMI and pointed out some data needed to establish and considered. And this includes sensitivity, specificity, precision, suitability controls, acceptance criteria, viability, and so on and so on. So the RMI assay for the oligonucleotides might be not very different one from for the peptide drug products. But when we design the assay, we need to be more careful thinking of the characteristics of these oligos. And I would like to focus on more sensitivity and stability controls in this talk. So we wanted to detect tiny small difference in impurities between RLD and generic products. So assay should be very sensitive. And it's really important to decide which cells you are going to use in your assay. You can use either PBMC or cell lines. Both platforms can be provided, but there is a pros and cons. So in the PBMC, so you can find a lot of literature saying that PBMC is more sensitive to RMI compared to the cell lines. And this is a mixture of different population of cells. And each population has a diverse receptor repertoires. So it can cover broad range of innate immune responses. And you can see, for example, like this, different cell types express different theaters. So it can capture broad range of impurities. Either frozen or fresh PBMCs are fine as long as the assay is sensitive. However, this PBMC has some there is limitations, donor to donor variation. So to get the statistical meaningful data, we need to test the blood at least from 20 donors, so it's a lot. And let's move on to the cell line. There are several cell lines you can use and some monocyte and macrophagic cell lines. They do have the reporter genes, so you can use easily. And this one, if you are using the cell line, the assay viability will be little, and you can do the high throughput assay. However, as I mentioned, this cell lines has lower sensitivity to RMI compared to the PBMC, and another challenge is coming from limited receptor expression for the nucleic acids. For example, THP1 cells, we are using a lot for the RMI assay, but THP1 cells do not express TLR3 and 9 at high levels. That means it can affect the assay sensitivity. This is our publication, and this study shows while PBMC are more sensitive compared to the cell lines. You can see overall PBMC, they respond to the lower levels of impurities compared to the cell lines. And if you look at here, I'm not sure whether you can see well, but THP1 and MM6, they are barely respond to poly-IC, imiquimab, and CpG, which is a ligand for nucleic acid. So if you're using these cell lines for oligonucleotide assay, you cannot capture the impurities, oligonucleotide-related impurities. So those are not very appropriate to use in RMA assay in oligonucleotide therapeutics. Understanding the impact of product on cell viability and responsiveness is also critical to assess the resulting data because that cells cannot recognize and cannot respond to the RMI. In the immunogenicity assessment for the generic peptide drugs, we sometimes see the problem. APCs, their main cells responding to RMI, they die in their assay. So they cannot capture the RMI. That is because of the toxic excipients such as phenol or polysorbate. They can affect to the cell viability. But we do not think oligonucleotide has this excipient problem because mostly the formulation buffer for oligo drugs are sodium salts. So we do not expect the impact on cell viability, but still need to check whether API and impurities can impact on cell viability. This can vary across the cell types, so it might be ideal to check the cell viability on each cell types. Once you set up the sensitive assay, it's also important to check if your assay can capture a broad range of RMIs. So we are calling that the stability control importance. So when you choose the stability controls, you need to think about whether it'll target a broad range of pattern recognition receptors, and also type of the impurities in your product should be considered. In oligonucleotide drug products, we expect some TRRs for the nucleic acids. They can be the big targets, so we should include the stability controls targeting nucleic acid sensors. But not only this, we should also think about the process-related impurities. They can target the extracellular pattern recognition receptors, so we also need to include some of them as a stability control. Then going back to our original question, can we detect RMI in oligonucleotide drug products? So to answer this question, we did some proof of concept study. And this is mostly done by Yan in our group. And we considered all the parameters I just mentioned, and we designed this proof of concept study. And to make a sensitive assay, we compared the human PBMC, THP1 dual cells, and also Ramos cells, which is B-cell line. We compared, and then we checked the response to the different impurities, and we found that PBMC showed the best response to the RMI. So we chose to use this PBMC. And as a proof of concept study, we synthesized ASO, which has PS backbone, 2 MOE modification, and 5' methyl C. And actually, its sequence is same as nusinersen, but it's just research grade. And we dissolved this one in the formulation buffer, same as the nusinersen. And also we included model impurities. We considered the potential impurities in this type of drugs. So we included several nucleic acid sensor ligands and also the extracellular PAM ligands. They can cover the various innate receptors. So to see whether we can detect these impurities in the presence of ASO, we put these in the fresh PBMC for 22 hours, and then we checked pro-inflammatory gene expression. First of all, we determined the highest concentration of ASO that do not interfere cell viability. You can check the cell viability with different assays, CCK, trypan blue, and flow cytometry. We use all of them, and then we found that these testing ASO do not impact on cell viability, as you can see here, and up to 1 to 10 dilution. And we move on to the model impurities. So we checked whether adding the impurities to this ASO can increase cell death. But as you can see here, even at the high concentration, you cannot see this model impurity, in this case CPG, did not increase cell death. So we rationale to use 1 to 10 dilution concentration for further assessment. This ASO, I mentioned that it has PS backbone, but it looks like it's not immunogenic. We checked several pro-inflammatory cytokine, pro-inflammatory gene expression, IL-6, IL-beta, and MX1, and it looks like there's no induction of those genes by ASO alone, and even at the highest concentration. So we spiked this ASO with CpG at different concentrations and see whether we can detect the signals. And this blue part, it's without ASO, and this part is with ASO at the highest concentration. And these are those dependent response by OND, the CpG. And you can see that there is a slight decrease of assay sensitivity here, but still the presence of CpG, even though there is an ASO, it could be detected. It's more than twofold induction compared to the ASO alone. So we can say presence of the impurity can be detected, but the assay sensitivity might be affected by ASO. And we move on to the other agonist. Since oligonucleotide can have a lot of oligo-related impurities that targeting TLR3, 7, 9, so we included those agonists as a stability control. And also considering the process-related impurities, we included the ligands for TLR4 and TLR26, LPS and FSL in this assay to see whether we can capture those responses in the presence of ASO. And you can see here, it's again, with the highest concentration of ASO, there are some decrease of assay sensitivity, but we could detect those impurities. The assay sensitivity, we can talk about a little more. Poly-IC, for example, without ASO, we could detect up to 1,000 nanogram per ml of poly-IC in our RMI assay, but with ASO, we could not detect by IL-6 expression levels. Interestingly, when we checked MX1 expression, we could detect it. But I'm not saying MX1 is the better marker for this assay, because if you look at this R848, you cannot detect here. If you go back to here, you can detect with or without ASO, you can detect up to 100 nanogram per ml of risankizumab was detected by RMI assay, but now it's not detectable when we check by MX1. So it means again, as Daniela mentioned yesterday, we need multiple biomarkers for RMI assay. We should not depend on just 1 or 2 markers. So in summary, modal impurities are detectable in presence of test ASO, and I cannot say all. Due to the limited time, I cannot tell all our research results, but it looks like the sensitivity to some agonists is reduced in the presence of ASO, but they can be detected. So in conclusion, a fit-for-purpose RMI assay may be useful in assessing whether generic oligonucleotide therapeutics has impurities that can cause increased inflammation or immunogenicity. The impact of each drug product on assay sensitivity should be established before doing the test, and the use of appropriate stability controls are very important to understand the breadth and sensitivity of your assay and interpreting your result. So I'd like to thank Daniela and Yan, Mohan and Seth, and our lab members to support this project. And also we have the collaboration work with Dr. Yang's group. So I appreciate, and also Steve, Susan, Mayumi, and Eric, Hobart, I'd like to thank. Yeah, thank you.
you, Dr. Lee, for a very nice summary. Now we come to the last but not the least talk of this session from Mr. Filip Kolenc. He will share some of the experience he has from developing a generic oligonucleotide drug. The title of his talk is Safety Consideration, Including Immunogenicity when Developing Generic Oligonucleotides.
Good afternoon. Thank you, Deyi, for the introduction. So today I will talk to you about certain safety considerations in generic oligonucleotides, providing an industry perspective on this topic. And also, perhaps as a toxicologist, also I will approach this topic from another way, let's say. So I will talk a lot about hazard exposures and doses and so on. So first, we will touch on the main differences between the generic and originator product, focusing on impurities. Then we will talk about safety considerations, general toxicity, and immunogenicity. And lastly, also touch leachables as a special type of impurities. So as you know, the preferred regulatory pathway for complex generics is the ANDA submission, meaning that when you demonstrate the sameness of the API through comprehensive characterization studies, no clinical trials are needed. Now, it's important to understand that the majority of the aspects between a generic and originator will be the same. So we will have the same API, have the same posology, treat the same patient population, and not also that, but we will even have the same manufacturing method, which is the solid phase synthesis. Nevertheless, the concern here is to have a different impurity profile which could compromise the safety of the product. And this isn't limited only to general toxicity aspects, but also to immunogenicity risks as well. So there could be impurities binding to the receptors of the innate immune system and leading to their activation. So let's start with process-related impurities. Now, compared to the API, they are much smaller in size, usually neutral or single-charged molecules. They can be reagents, starting materials, solvents. Usually we control them according to existing ICH guidelines, but more importantly, due to high purge factor, they are effectively removed and not present in the final product. And this was very nicely demonstrated by Filip and colleagues in a recent article, where they demonstrated that even for potentially mutagenic impurities, they are basically cleared by the process and not present. And this just emphasizes how high, how robust this clearance of process-related impurities by SPS is. So in conclusion, these impurities are cleared by the process, and also to add, small molecule impurities are mostly non-immunogenic. Now, an exception to these are leachables because they can form downstream of the purification process, they can form during stability. That's why we need to take a closer look at them, and we will talk about them later. So let's now shift our attention to product-related impurities. Now, they can form due to incomplete or side reactions. They can be degradation products. Some typical types of impurities are abasic sites, N-1 deletions, or N+1 insertions. Although they are more difficult to remove than process-related impurities, it's important to know that chromatographic techniques of separation will generally remove all the similar impurities to the API. So, for example, in our experience, N-2 and N+2 impurities are already sufficiently different by their physiochemical properties, in this case charge, to be mostly removed, and we do not see this in the product. So if we put this into one chart, what we can see is that some of the impurities have low safety risk. So even if there is a potential hazard, there won't be any exposure. On the other hand, there could be some similar to API impurities present, and often the risk is unknown, and that's why they require a closer inspection. And one thing that we can do now is to categorize them into low risk and high risk safety groups. And one way to do this is by applying a classification system, as was proposed by the Oligonucleotide Safety Working Group, and also recently adopted by EMA in their quality guideline. I think it's from July this year. So this system contains 4 classes. Class 1 and 2, such as major metabolites, are not considered to pose any safety concerns. However, there could be some sequence variants of the parent, or there could be impurities containing structural elements not found in the parent that could pose some safety concerns. Now, it's important to note that this classification system, as was proposed originally by innovator companies, has found acceptance among some regulatory bodies. However, in our experience, not all regulatory agencies might accept this approach for generics. That being said, let's continue with the general toxicity aspects of these impurities. So when we talk about class 3 and 4 impurities, it's important to understand that these are mixtures of components of the same structural group, so-called positional isomers. And analytically, it's very difficult to resolve individual impurities and to demonstrate that you have the same impurity profile. So for example, we might have the same level of A-basic impurities as in the reference, but the ratio of individual might vary. And what this means is that we would typically need to perform an impurity qualification study in animals. However, there is an alternative possible, which is the application of sequence-independent impurity toxicology. And this is a concept that was proposed by the Safety Working Group. And basically what it means is that within one such type of impurity, for example, A-basic, that regardless of where the base is missing, the physiochemical properties will be similar. And by extension, they will also have similar toxicological effects. So this means that studies would not need to be repeated to qualify the same impurity type as already contained in the reference product. Now, this concept is supported by some published impurity qualification studies in animals, where they tested APIs highly enriched with impurities like 50% API, 50 impurities, N-1 total, A-basic total, for example, and these did not contribute to observed toxicity. So in conclusion, further research on this concept would be very valuable and could reduce unnecessary animal testing. And if I heard correctly before from the presentation of Kui, I think this is actually ongoing. So let's now switch our focus to immunogenicity considerations. So the concern here is that there could be a deletion, insertion, or substitution of a nucleotide, which might create an inflammatory sequence motif. So if you look at the literature, we can see a lot of sequence motifs mentioned, but the most widely recognized and potent is the CpG motif. So let's take a closer look at this motif to understand what kind of risk it poses when present in impurities. So as you are well aware, bacterial DNA contains many repeats of unmethylated CpG motifs, which can induce an immune response in mammalian systems. And this motif was also used in vaccines to improve antigen presentation and facilitate an immune response. And example of this is the CpG adjuvant 1018 here on the slides, which, if I'm not mistaken, is the only FDA-approved CpG adjuvant for human vaccine use. And it works by activating toll-like receptor 9. Now, it's important to understand that besides the PS backbone, this adjuvant is completely unmodified. It has no modified sugars or modified bases. And this will actually never be the case in generic oligonucleotides or by extension in its impurities. And as we will see on the next slides, modifications are key to reduce an immune response also for impurities. So firstly, as you know, methylation of the cytosine in the motif demonstrates a lower binding affinity to toll-like receptor. And if you look at all the approved ASOs, we can see that all of their cytosines are methylated. So it is very unlikely for an impurity to arise which would be completely demethylated. But more importantly, some azoles, which are, let's say, not vaccines, already by themselves contain CpG motifs. So again, just looking from a toxicological perspective, if there is an impurity present that has a CpG motif, it wouldn't really add much to the risk because you already have a high dose CpG containing molecule present. Secondly, and most importantly, sugar-modified nucleotides reduce TLR9 stimulatory activity even without methylation of the motif. And this was nicely demonstrated in a recent article by Yoshida and colleagues. And again, if we look at most of the approved therapeutics, we can see that they are mostly sugar-modified, and some of them are even fully modified. So again, if there would be an impurity, CpG containing impurity present, its potential immune response would likely be mitigated by the presence of sugar modifications. And I think they also shown that on the neighboring nucleotides, if, for example, they reduce the response. Thirdly, to really have an impurity present which would be able to strongly activate human TLR9, you would actually have to have two closely positioned CpG motifs as well as optimized flanking sequences. And this is actually what we can see on the previous adjuvant. So we can see it has two closely positioned CpG motifs as well as optimized palindromic sequences. And it is highly unlikely for such an impurity to arise, if not even altogether impossible. And also for those azoles that do not have a CpG motif by themselves, this would actually require the generation of an N-2 or N+2 impurity, which, as mentioned before, would be basically removed by chromatographic separation. So lastly, let's also look at the doses. So even optimized CpG oligonucleotides require doses above 5 milligrams to exhibit robust pro-inflammatory effects. So we can do a short, let's say, quick calculation. We take the doses of most of the approved therapeutics. We adjust for human weight, and we apply the qualification threshold of 1.5%, which was proposed by Safety Working Group and recently adopted by the EMA in their quality guideline. And what we see is that in most cases, we are below this threshold. So even if there would be an CpG containing optimized impurity present, it would still not be present in such high amounts to be able to exhibit some robust effects. And in those cases that are above, it's important to note that they are all fully PMO modified therapeutics. And this modification is well known to not lead to any kind of immune response. And because they are fully PMO modified, their product-related impurities will also be fully PMO modified. So in conclusion, impurities will not be present in such high amounts to be able to exhibit a robust pro-inflammatory effect, nor will they have optimized sequences. And so the overall conclusion here might be that the inflammatory motifs in impurities might not really represent a concern. Let's also touch quickly on the complement activation, as also mentioned before. So this is also a component of the innate immune system. As was mentioned before, PS-modified oligonucleotides can elicit the alternative complement pathway due to plasma protein binding. However, there is indication that this is a threshold-dependent effect. So doses as high as 6 milligrams per kilo did not produce a significant or consistent complement activation. And again, we can do a quick calculation. So we take the doses of most approved oligos, we apply the qualification threshold, and we see that in all cases, we are well below the threshold of 6 milligrams per kilo. And that is also true for the, I think, the high dose Viltolarsen, which I think can be given like in a few grams. Now, it is true that this threshold pertains to 2-hour intravenous infusion, and this might not account for, let's say, a worst case scenario where the patient would get like a bolus injection. However, nonetheless, due to low plasma concentration of modern ASOs, impurities are also unlikely to exceed this activation threshold. Also, let's touch shortly, before I go to the next topic of leachables, on some challenges. So what we can do is similarly as for peptides. So you can demonstrate the lack of immunogenicity by performing in vitro assays with the product. So you take your product, you incubate with PBMC cells, and you measure a pallet of cytokines. And you do this over the entire product shelf life. However, in a hypothetical case that you would get a positive result, the problem is that you have no way of knowing which impurity to reduce. So if the analytical people come to you and ask you which impurity to reduce, you don't really have an answer. And we have had a similar discussion yesterday on the small group discussions. And one of the problems here is also that you cannot test individual impurities due to significant analytical challenges, such as separation of positional isomers. So here I think an opportunity would be to develop studies to be able to pinpoint the specific impurity responsible for triggering potential immune reaction. And I think that we got good indications in the last talk by Hannah. So lastly, I would also like to talk about leachables. So they are small molecular impurities. They are many times called as contaminants, and they can come into the product by leaching from the filters, plastic tubing, needles, stoppers, plungers. And usually what we do is we control them according to PQRE and LC recommendations. So we check whether or not they are mutagenic. If so, we apply the TTC limit. And we also check whether or not they cause sensitization reactions, if they have sensitization potential. And if so, we apply a threshold limit of 5 micrograms per day. And interestingly enough, this is actually a regulatory accepted immunogenicity threshold. Nonetheless, leachables cause an additional risk. That is that they may react with biomolecule drug substance, compromising safety and efficacy. And in a recent discussion with a certain regulatory agencies, it was highlighted that there could be reactive leachables present which could engage the RNA target, your API, which would then in body recruit RNases, cause the degradation of the API, leads to the increase in product-related impurities, and they could potentially lead to immune reactions. So, interestingly, this evaluation of potential interactions with leachables was also highlighted in recent EMA quality peptide and oligonucleotide guidances. So, naturally, one asks himself, what is a reactive leachable, and can we predict interactions with RNA? And as a matter of fact, it seems that we might. So this was nicely demonstrated by Johnson in a recent article where they used a combination of in silico approaches to address the possibility of leachable biomolecular reactivity, and it was determined to be a fast, effective, and conservative approach. And also, when I was thinking about this topic from toxicological point of view, I was actually surprised because, in a way, this is very similar to what we already do in the scope of ICHM7 evaluation. So, in both cases, you have a small molecular impurity binding to nucleic acids. So, perhaps a good starting point would be the use of already, validated and regulatory accepted QSAR tools, which are used within the scope of ICHM7 evaluation to really determine these hazards of leachables. And this could ultimately lead to their eliminating any safety risk they pose. So in conclusion, process-related impurities seem to be removed from the manufacturing due to high purge factors and thus pose low safety risks. Product-related impurities would benefit from being able, to categorize them into safety classes And the application of sequence-independent toxicity would allow or reduce animal testing. Inflammatory sequence motifs, at least for the CpG, seem to be unlikely to represent a realistic safety concern for impurities. And also complement activation seems to be dose-dependent, with a threshold impurities are unlikely to exceed. And also reachable API interactions seem that could be determined, identified by already available QSAR tools. And the challenges and opportunities I've already talked about during the presentation, so this is all from my side. Special thanks to the Sandoz team that is working diligently on oligonucleotides. It's a very challenging topic. And also thank you for this opportunity to be able to speak today.
Thanks, Filip, for sharing your opinion, also considerations from the generic company point of view. Now I want to invite all the speakers to the podium. Remember, we also have 2 virtual speakers. I'd probably first give our speaker a round of applause for their very nice presentation. So for the next 30 minutes, we will have a panel discussion. This is the only session for synthetic oligonucleotides. So if you have any questions, don't hesitate to ask.
Thank you very much indeed. I have a question for Dr. Lee, just in relation to the selection of the ERMEs for the oligonucleotides. Towards the end of your presentation, you mentioned that it's going to be quite important to select a wide range of different ERMEs, obviously depending on the specific impurity types in the oligos. In the context of validation of those assays, then obviously that means that there's going to have to be quite a wide selection of ERMEs that go into that sort of validation step. 2 parts. Can you kind of comment, or do you think the FDA is going to be kind of providing, I guess, are you going to be publishing this data in relation to highlighting more details in relation to those individual ERMEs? And in particular, the particular cytokines, chemokines that you would be wanting to, that from your data would suggest it's kind of good to look at. And also, from your own experience, the challenges of validating a wider selection, a wider panel of different ERMEs. How practical is that going to be, do you think, and where are those issues going to be facing?
Okay, so I think first of all, FDA do not suggest or recommend certain strategies. So you can bring and then we can review and then we can discuss together which way is better to review. But as Daniela mentioned and Seth also pointed out, we need to think about the impurity characteristics and but we can fill our plate with all the impurities, right? We can test all of them, but as much as you can cover all possibilities, it'll be acceptable. And what was the second question? I couldn't get it.
It was more to do with just the practicalities of validating that, because obviously in that wider selection, it's going to be very much driven by those impurities. And at this stage of everybody's experiences, just to ensure those right characteristics and right ERMEs are selected.
I think FDA and industry, we all learning about this new field. So I have right answer, but if you have more questions, you can discuss with Daniela. She has more idea about that. Sorry.
Thank you.
If I may also, with regards to impurities, I think we learn a lot about these during the manufacturing process, even for the new drugs. And when the new drugs switch their facilities or they're scaling up, you're going to see some of these changes as well. And so as we learn from those, we can then inform our generic environment. Of note, the test versus reference issue is very different for especially if you're looking at multiple tests and multiple impurities, it gets the difference detection ability, you start dropping quite rapidly the more things you test, because then and there are various reasons for that, including statistical and just the whole issue of practicality.
Daniela?
I just wanted to to try to answer Jeremy's question, because the first part was one that I think some people are hoping for, which is, are we going to issue some guidance? And I think that it's going to take us a while. I think that we're still learning in this space. And so I think Hannah mentioned that there's a lot of PSGs that we're putting out, and we're saying consult the agency. And when you consult the agency, a lot of times you're going to get, this is our advice for now, but our knowledge in this space is really evolving. And that's where we are. And I think this is a wonderful meeting to learn from, just heard Filip saying, no matter what you do, no matter what impurities you have, there's absolutely no concern. I don't know how you go from there to, products we have 50 or 70% immunogenicity, but that's his stance. That's and we're learning, and we're taking this information and we're putting it all together. And we heard from Sudhir, just a masterclass in oligos and the risk they pose. So again, we're putting it all together, and hopefully, with the help of all of you, we'll have some guidance soon.
And Daniela, we're not alone, right? Because we have our friends in Europe and the EMA folks as well, and they're learning from folks in Slovenia as well as we are. So.
I have a question for Dr. Rogers. One of your slide, you mentioned that some of the oligonucleotides NDAs are approved under subpart H of CFR. I believe that means there are no complete clinical trials were done for those oligonucleotides. If that understanding is correct, so whether we will be allowed to file ANDAs for those NDAs which were approved under subpart H, and since they were approved under expedited approval program, whether our ANDAs will also be approved under expedited approval program?
Yes, hopefully you can hear me. So I want to clarify, there were clinical studies done. They were not approved. They were approved on the endpoint of dystrophin for the muscular dystrophy drugs. Also, they're waiting for a confirmatory endpoint on a hard clinical endpoint. So to clarify, so clinical studies were done.
Okay. That's good to know. Thank you.
First, I'd like to thank the panel for beautiful talks. And my question is for Dr. Agrawal, but also for the panel in general. For the PS linkage, like you showed us nicely, that when you incorporate it, it does lead to the formation of like 2 to the whatever power diastereomers. And we saw that it impacts their nucleus stability and their immunogenic response, and also their hybridization properties to the target mRNA. And we see all of these like properties change in vitro, but my understanding is that that does not necessarily translate to animal studies. So for example, if you have like different distributions of these stereochemistries, they will have different in vitro properties, but that doesn't necessarily mean that one diastereomer distribution will have more efficacy as compared to the other in animal models. So I was wondering if you could highlight what would be some of the factors that would result in this disparity.
There are 2 isomers, S and R. So R isomer is more like a natural DNA, and S isomer is more stable. So that affects the protein binding. Protein binding will affect position in animals. Then the stability will be dependent. So if its position is different, then R isomer will be degraded faster compared to S. So you can imagine injecting a mixture, and slowly R isomer is degraded. At the end of the day, it's the S isomer which is residing in the body.
That's just for that one instance. I mean,
Yes.
every oligo is different.
Yes.
Don't take that as a general rule.
Okay, here's a question from the online audience. This is for Dr. Kui Yang. For impurities profiles in oligonucleotides, how much MS fragment ions needs to be to match in the sequence coverage?
So the target will be 100% sequence coverage. But if you are not able to get all the fragment ions that can give you a 100% sequence coverage at the first time, you can try using, like I showed in the presentation, by optimizing the collision fragmentation technique, collision energy, and also you can try using different charge states. And if you have to use more than one charge state to get the fragment ions that can give you a 100% coverage, that also is applicable. If you have exhausted all the potential ways in technology to give you 100% coverage, if 100% coverage is not applicable, then we would like to see the justification to show how those less than 100% coverage is not affecting the safety and the quality in your review package. Yes, so in short, the target still will be the 100% coverage.
This is regarding the abasic impurities in oligos. Given their unstable nature, what is the relevance of testing these abasic impurities in toxicity study?
Want to try?
That's a pretty general question. So oligonucleotides, you're saying they're unstable, so why study them? I think some of them are more stable because of our man-made constructs. I mean, your basic oligos with just regular, like the regular CGA, et cetera, those are going to be unstable because your RNase will target them and knock it out. But the ones that we're looking at, the man-made constructs, are relatively more stable.
I'm talking about abasic sites, right? Abasic sites and abasic impurities, which are unstable.
Filip, you want to comment?
Yeah, I mean, that is an interesting question. Yes. So at the moment, it's just purely, I guess, what the agency wants to see that, from a toxicological point of view, you qualify these impurities, whatever they may be. So these animal studies that were made and published in the literature, they used all common impurities they have in the product, including abasic. So they had abasic total. It was a mixture of abasic impurities in ASOs. And it seems that at least in the product, it's stable. Otherwise, they wouldn't test it. So, yeah.
I think just to add, a basic site in a natural DNA will be degraded, but in these cases, a basic site is between where the two flanking phosphodiesters are modified. So they're not substrate for you.
Thank you.
Before I go to online question.
Okay, this question is for Filip Kolenc. What was the max size of oligos tested for immunogenicity? Because there are more impurities as the length increases due to additional synthetic steps.
Yeah, sorry, I heard the first part of the question, but not the second, sorry.
Okay, let me repeat, sorry. What was the max size of oligos tested for immunogenicity? Because there are more impurities as the length increases due to additional synthetic steps.
Yeah, I mean, it's still, between what currently there is. So 20 to 40, these are the oligonucleotides of length that are available for generics. And you might have a n+1 impurity, I guess, but the n+2 impurity would already be separated. So that's the longest, I guess, that would pose a risk.
Okay.
Yeah, this question regarding the qualification thresholds. Are we adapting the EMA guidance?
Filip, maybe?
No, we don't have an interagency cooperative on that one yet.
You are talking about qualification threshold, right? Yeah, do you want to comment?
So, I mean, at the moment, what we see is that each agency currently has its own thinking. And what I would maybe highlight is that at the moment, the ICH S13 guideline on non-clinical considerations for oligos is now being developed, kicked off recently. And one of the things that they will also discuss is impurity qualification, reporting identification threshold. So I would say that maybe in the future, the next years, there could be more clarity on this and perhaps also some harmonization.
I know there's a white paper, right, widely cited in the oligo field. But as a regular agency, we don't really simply just accept those suggested thresholds. So it's kind of a case by case. So you need to really analyze your own test product and see what impurities you have and compare with ILD. So it's not like simply 0.5 or 1%.
Hi, this is a general question. From what I've heard so far, it looks like thrombocytopenia, complement activation, generalized inflammation, and development of anti-drug antibodies are the immunogenicity risk factors here for oligonucleotide drug products. Do we know of the source or what in the drug product contributes to these factors so that we can better control them? Do we have an experience on the specificity of anti-drug antibodies? Because we see like 50-60%. Do we know what it is against? Complement activation and thrombocytopenia. Do we know if there are macromolecular complexes that are formed in vivo? Do we know the source of these issues so we can better control them? Generally to the panel, anybody who is willing to take it.
This is Bart. I can speak to an extent. For the thrombocytopenia, we do know that tends to be a dose-mediated phenomenon with PS backbone oligonucleotides. Systemic doses, typically around 250 milligrams to greater than 300 milligrams weekly, were cause of that tended to be perhaps both an IgG and IgM-mediated phenomenon. There were some non-human primate studies done there for the cause of thrombocytopenia. As to the other ADA phenomenon, ADA positivity, that it does depend. If you'll notice in my slides, in one case it was IgG. It varies only on the specific assay there. So that phenomenon, we're not still exactly sure. We have to look into it further.
So I have a question for the entire panel. One's a bit of fact checking on me and assumptions, and then I'd like to hear your comments. So we learned TLR3s are responsible for binding double-strand RNAs. So that would be siRNAs kind of fall into that class. That receptor is absent on human B cells. So that's the fact check part. And then assuming that's true, I wonder if that has any bearing why we haven't seen appreciable ADA, at least to my knowledge, for siRNAs. And then contrast that with what we heard about with ASOs. And it's interesting, of the ones that have robust responses, they all seem to have a very common 2' MOE modification. So have you seen that at the agency and or experts, and then comment as what might be driving that phenomena?
Just to address the nature of adverse events for these products. We pointed out early that many of the indications are rare, so you can have very small populations using these. And so depending on which drug we're talking about, some of them are going to have a larger population, some of them are going to have a smaller population. So the fact that you see any adverse events for some of these is going to be less likely. And again, it's a risk versus benefit approach. We're looking at the overall risk of the product and the fact that even in the new drug arena, for the RLDs, they're going through multiple batches that are relatively small. And so you're going to see maybe some variation from batch to batch there that we may not be able to capture as well because with regards to potential impurities, et cetera, simply because of the small populations that are being targeted for these products.
Well, I understand the small population and adverse events, and that could be innate driven, but if we focus on treatment emergent anti-drug antibodies, that's kind of where the basis of my question was going in terms of the lack of TLR3, and so siRNAs seem to be safe, and perhaps by analogy impurities, and then the observation that of the ASOs, again, they all seem to have the 2' MOE, and is that driving the treatment emergent immunogenicity to those particular molecules?
I think it'd be interesting to watch the population and see what happens in oligos, but there are some lessons learned from the big molecules, from the BLAs, right? We've seen that young women are more likely to develop anti-drug antibodies early on, but the overall men versus women, they get it becomes aligned and there's no difference there. And certain products are more likely to develop ADAs than others. We're not sure if that's because of impurities or not. So I don't know if folks online might want to chime in.
Yeah, I can speak to an extent. So go ahead, Dr. Agrawal.
I think for TLR3, look at early stages of siRNAs which were less modified. They were activating TLR3, but since they are heavily modified, the receptor doesn't recognize those molecules anymore. They're not present on human B cells, but in general, just to answer, so TLR3-based agonists have been used as vaccine adjuvants, so they do lead to antibody-mediated immune responses. Early candidates in siRNA were less modified, so they were activating TLR3. That was one of the mechanisms of action which was questioned. But since then, they have been heavily modified, so they do not activate TLR3. Bart, you were saying? Yeah.
Yeah, I'll just add to that. I mean, we have seen anti-drug antibodies in the approved siRNAs, although, albeit it's much lower to an extent, but I don't know if we're, still out of the woods and saying that, there's not necessarily activation. There could be other things going on. In addition, we do see injection site reactions with those products typically when given subcutaneously. So there's still things to be investigated from that standpoint for sure.
Yeah, my question is for Hannah Lee. You mentioned about animal immunogenicity studies, comparative immunogenicity studies in animals, where at least it tells you whether your product has more immunogenicity than RLD and so on. However, there are so many instances for many of the biosimilars that are approved, whether it is monoclonal antibodies or a fusion protein, where animal immunogenicity studies have given much higher immunogenic responses. But when the same molecules have gone into clinic, it's just the opposite. Is this something that has become quite often? There are about 8 to 10 such instances. Shouldn't this comparative animal immunogenicity studies be discontinued because it doesn't serve any purpose?
Yeah. So actually in my talk, I did not talk about the animal study, but according to the today morning talk, it looks like we can make animal studies can mimic what we observe in clinical, like there are two possibilities. One is humanized mice and also tissue explants models. But I understand, yeah, so immunogenicity risk in mice and human totally different. And also not only just like RMI, like innate immune modulating, the part also complement activation part, there are a lot of debating. Like sometimes usually the sponsor bring the justification, they do not think complement activation assay is needed because one group published several papers that monkey is more sensitive to the impurities, so complement activation can easily happen, but not in human. But there's another also like opposite observations from other companies that human complement also can be activated. So we are still, like, again, we are still learning about this new field. So maybe if there's any better models to predict immunogenicity risk, it would be awesome.
But it's also comparative immunogenicity, right? The test versus reference. That's a additional hurdle.
Okay, another question from online. Can an oligonucleotide from a well-characterized chemical class forego immunogenicity testing?
I can speak to that. No, currently not, because I think as you've seen in the presentations today, even subtle changes can greatly influence the immunogenicity risk for these compounds. So changing the backbone or change the sequence of the RNA bases, even if you're using a well-characterized chemical class, can certainly impact immunogenicity. So it does not forego the need for immunogenicity testing.
We'll probably go with another online question.
Okay, this question is for Filip again. Are there any in silico approaches for evaluation of oligonucleotide immunogenicity or inflammatory risk assessment?
So, as I understand, if there's any equivalent, let's say, to in silico approaches that are used for peptides, right? So, at the moment, not that I am aware of. So, the only in silico approaches in terms of oligonucleotides would be more like the analysis of off-target effects, but that's not immunogenicity related. So, not that I'm aware of.
Okay, so for the interest of time, we just have one more question. Do we have any in-house? Okay.
With no guidance around the impurity thresholds for qualification or anything, is there anything indicative, or it is simply based on the, or any LOQ that the agency suggests that at least should be targeted by the methodology being used? Because otherwise, comparison to what level becomes a challenge for anybody who's developing these products. Test versus R&D.
So you probably get the response already from the agency, right? From the PDAF meeting. Typically, you just compare with the LD. I mean, if the level is lower than the LD, safe. But if you have high level of new impurity, depend on what kind of impurity you consider, immunogenicity, also maybe tox even. So,
yeah.
But if you have specific question, you can always request a meeting or use a control process.
Thank you.
Same or lower, right? And we also know that you can tell by looking at the RLD, doing reverse engineering, thinking about how that product was made, what kind of product synthesis it went through. There, for example, an enzymatic approach versus some other approach to making those oligos.
Thank you.
Okay, so do our online speaker have any comments on that? No? Okay, that's okay. So I think, it's time to wrap up the panel discussion, and let's give our panel another round of applause. Thank you very much.
meet you. Thank you for flying all the way. Go here.
Next, I want to invite Dr. Rob Lionberger, Director of Office of Research and Standard in the Office of Generic Drugs, FDA, to give a closing remark for the virtual attendance. Thanks.
Thank you, Deyi. Fantastic meeting. I really want to thank our organizing committee who put together the program, our CRG staff who helped get all the logistics working out, all of our speakers who traveled here to participate and engage in person. It's a fantastic meeting. And, but it's not just fantastic, it's also incredibly important. And I just want to remind everybody here in the room and online how important this topic is to the Office of Generic Drugs. You heard our office director, Elin Murphy, open the meeting, and the reason she was here is just the incredible importance of generic competition in the peptide space is to the American public, to the generic program, to the generic drug industry. This is a critical issue, right? You heard many people on the slide say, look, in 10 years, there's going to be a $150 billion market for GLP-1 peptides. This is a huge access to these products is going to, if the promise continues, the access to these is going to be a huge issue for public health. Even right now, you see drug shortages. People at this meeting told me, oh, a family member was prescribed this, but they couldn't get it. Maybe, can I try the compounded product? I think that's the reality. If we are not all in and all on board and really focused on making generic competition available for these products. The generic versions, they go through the rigorous FDA review, the full supply chain, everything, the level of quality in those products is the same as the brand product. And we have to do everything we can to make sure that we have a strong, efficient generic program for competition in these areas. It's probably the most important thing the generic drug program is going to do in the next few years. And I want to make sure all of you listening and thinking about that recognize that. I think from the industry point of view, this is opportunities to contribute to public health on a massive scale. For those of you who work for FDA who are here listening, really important that we are really moving, aggressively, thoughtfully, but emphatically toward that goal. And I just want to give a little bit of just my perspective of 20 years in the Office of Generic Drugs on some of the aspects that are characteristics of a highly effective generic drug program. We know from small molecules, generic drugs save American public hundreds of billions of dollars a year in pharmaceutical costs. But why does that system work? And I think reflecting on that gives us good directions for how we want to make sure our programs in peptides, larger peptides, and oligonucleotides in the future evolve so that we also get those same benefits in these other therapeutic areas. And so some of the features that are important is, when I think of the generic drug program, one of the things that makes generic drug development more efficient is you don't have to repeat unnecessary clinical studies. And this really puts the emphasis and importance on understanding the pharmaceutical science and the clinical pharmacology to say, we know why these drugs work, we know what the critical attributes are, and if we focus on those being the same, then we don't have to repeat the clinical studies. And also, I think you hear sometimes at this meeting, well, the brand company did a clinical study, so they didn't need to characterize the immunogenicity risk. We also have to recognize the limitations of clinical studies. Those clinical studies are not powered to find rare immunogenicity risks. They're powered to demonstrate, clinical efficacy in the population. And so, from the pharmaceutical science, clinical pharmacology approach, even for brand products, is a much stronger way to minimize risks for those products as well. So, certainly, for efficacy, the clinical trials are really the key aspect. But safety issues, much more challenging, especially if they're rare events. One aspect of the generic drug program I want people to see is very important is our product-specific guidances. And for this area, we want to, I think we aspire to having our product-specific guidances really give you the roadmap to the type of studies that you'll need for each particular drug product. right? What the risk assessment is, right? Do you need a full set of immunogenicity studies, or is this peptide a low-risk product? You have to be able to look at our product-specific guidances and see that. And if you look at other areas, our bioequivalence studies in the generic program, we get hundreds of applications every year. Almost all of them are first cycle correct because we have product-specific guidances that very clearly tell you in those PK studies, what's the study design? What analytes do you need to measure? All of the key characteristics that let you get the studies right the first time. And that's our aspirational goal for this area, to really have the immunogenicity assays, that level of reliability where you look and say, for this product, these are the ones that I need. Everybody knows how to do them, how to validate them, and you can have an efficient development process around them. And if you think about future competition, if you want to have multi-source suppliers, you have to have a system that allows different manufacturing sources. If you want multi-source suppliers, you have to have that. And that recognizing that there have, so some of those differences in manufacturing process. I think in the peptides, you see it most clearly in the difference between recombinant sourced and synthetic sources, that those manufacturing processes, they produce exactly the same APIs, but the impurities can be different. So if you want to have a functional, competitive generic system, you have to have a way to qualify, mitigate, manage the risks of those differences in impurities in some way. And that's a key goal for this. You heard the discussion here around moving toward the ability for generic sponsors to use the recombinant synthetic methods in generic drug applications. I think that that's going to be a necessary feature of the future state. And we want to really focus on what do we need to measure and compare, and building out the tools and making them reliable so that we can do that. And so again, also want to make sure that as you're considering generic drug development, you look at all the ways that you have to interact with the generic drug program through our pre-ANDA meetings and control correspondences to get clarity around this. Again, I said these are one of the most important classes of products. So please use those meetings. We want to grant them. We want to engage with you. We want to have scientific discussions. We want to give you feedback on innovative approaches. We also want to help you with implementation challenges as well. So please think about how you can use those meetings and interact with that so that your applications are right the first time. And again, just as a final closing remark, really appreciate today's programming, appreciate all the people who came here with the different approaches and experiences with these analytical methods. It's really important that we work together to really understand how to implement these, what the best practices are, and look at ways that we can share them. And if you want to go further on this, I encourage you to reach out to for the generic industry. One of the purposes of the Center for Complex Generics is to help facilitate collaboration between the generic industry through incubating consortiums, working with other groups to develop collective action where you can get industry input into making and developing best practices, input. This also helps FDA develop guidance when we get input from industry that's consistent and covers multiple stakeholders to look at ways you can develop and work together through the Center for Complex Generics to help us get to this final state. So with that ends the virtual part. For all of you in person, we'll be continuing the discussion. I think we'll be having a break first, but thank you all online for attending, and hope to see you at other CRGC events.